Good afternoon, students. Today we are going to start with the chapter Lesson 8 Cell Structure and Functions. In this chapter we are going to discuss discovery and history of cell, instruments for studying cells, staining, unicellular and multicellular organisms, level of organization, cell shape and size, parts of the cell, prokaryotic and eukaryotic cell. So students, let us begin with the chapter. Now there are millions of different things in this world around us. Some of them are living and some of them are non-living things. Plants, insects, frogs, human beings, etc. are living things, while sand, soil, air, water, chair, etc. are non-living things. Living things carry out all basic functions like respiration, digestion, excretion, reproduction, movement, growth, etc. Though non-living objects may perform one or more of this process, like vehicle can move, but it is only living things that performs and exhibits all the basic functions. Among living organisms, these are broadly classified into plants and animals, and all these organisms look different from each other. However, all of them are made up of microscopic unit called cells. In this chapter, we are going to study about the basic structural unit of living organism, which is cell, its parts, and the function of various parts of the cell. So students, let us study cell, the structural and functional unit of life, in detail. But before that, let us learn discovery and history of cell. An English scientist, Robert Hooke, observed the slices of cork under his self-designed crude microscope. Cork is a part of bark of a tree. He took thin slices of cork and observed them under the crude microscope. He noticed a large number of compartments in the cork slice, resembling the structure of a honeycomb. Robert Hooke coined the term cell for each box, which in Latin means little room. Robert Hooke had actually seen the dead wall of the plant cell. This cell had lost their living contents and looked like little room. Now students, let us learn about some instruments for studying cell. Cells are microscopic in structure and cannot be seen by naked eyes. Today we know a lot about cell structure and its function because of improved microscopes having high magnification. So first is compound microscope. Compound microscope is used in biological laboratory to observe tiny cells. It uses glass lenses and visible light for observation. Second is electron microscope. Is a very large instrument that helps in observing various internal parts of the cell. It uses electromagnets instead of glass lens and a beam of electrons instead of light for observation. The highly magnified image is obtained on a photographic film or screen. Magnification of over 1 lakh times may be obtained in this electron microscope. Staining. Cells are transparent and colorless. Transparent materials, which are also colorless, are difficult to observe under a microscope. We use colored stains, or we can say dyes, to observe details of the cell and its fine structure. Some common stains are safranin, eosine, methylene blue, and crystal violet. Now to observe onion peel cell under a microscope, compound microscope, let us go through this activity. Apparatus needed: an onion, glass slide, cover slip, a compound microscope, dropper, forceps, water, knife, watch glass, methylene blue solution, which is a stain. Method: now cut a slice of an onion and peel off its skin using forceps. You can see in image. And place it in a watch glass containing water. This will prevent the onion peel from getting dry. With the help of a dropper, put a drop of water on a clean slide. Place the thin piece of onion peel on the water drop. Put a drop of methylene blue on the onion peel to stain it. With the help of needle, cover the onion peel with a cover slip, taking care that there is no air bubbles in it. Observe the slide first under a low power of the microscope. For better details, observe the slide under a high power of compound microscope. Now what you are going to observe? You will see, while observing the slide under a microscope, you observe that a number of cells lie side by side. The cells are rectangular in shape. Each cell has a distinct wall. This is called cell wall. A prominent means important nucleus is present in the cell. A large empty space exists within the cell, and this empty space is called a vacuole. So from this, what we have concluded, like onion, all plants and organisms are made up of cell. Now organisms are of two types, that is unicellular organisms and multicellular organisms. Now let us learn this one by one. So first is unicellular organisms. Organisms made up of single cell are called unicellular organisms. For example, amoeba, paramecium and euglena. Bacteria and yeast are few examples of unicellular organisms. All the basic functions of a living being like respiration, excretion, reproduction, etc. are performed by one cell. Second is multicellular organisms. Organisms made up of many cells are called multicellular organisms. Most of the plants and animals that we see around us are multicellular organisms. For example, trees, grass, insect, cows, birds, humans are few examples of multicellular organisms. All multicellular organisms, including human beings, shows division of labor. It means different part of multicellular organisms perform different function. For example, human body has a stomach to digest food, heart to pump blood, skeleton and muscles to perform movements, and so on. Now students, let us discuss about cell size. The cells and their parts are measured by fractions of millimeter, that is mm. 1,000th of millimeter is called macron or micrometer. 1 macron or micrometer is equal to 10 to the power minus 3 millimeters. Most of the cells are microscopic in size and are not visible to the naked eye. They need to be enlarged or magnified by a microscope to be seen. The size of cell varies from very small cells of bacteria to the very large cell of eggs of ostrich. In our body, red blood cell is the smallest and the nerve cell is the largest cell. The algae, acetabularia, that is unicellular organisms, consists of a single cell having a length of 10 mm.